DNA replication. In this chapter, we will analyze the process and enzymes involved in prokaryotic DNA replication. You will learn how DNA is duplicated, the roles of various proteins in this process, and how prokaryotic and eukaryotic replication differ, especially in handling telomeres and chromatin reassembly. DNA replication is a semi-conservative process, meaning each daughter molecule consists of one parental strand and one newly synthesized strand. This process begins at the replication bubble or fork, and was first confirmed by the famous Mendelssohn-Stahl experiment, supporting the Watson-Crick model of replication. In prokaryotes, DNA replication proceeds bidirectionally, forming a theta structure. One strand, the leading strand, is synthesized continuously in the 5 to 3 direction. The lagging strand, however, is synthesized discontinuously as short Okazaki fragments because DNA polymerases cannot work in the 3 to 5 direction. Okazaki fragments are initiated with short RNA primers. DNA polymerase III extends the DNA, while RNase H and DNA polymerase I remove the RNA primers and fill in the gaps. Finally, DNA ligase seals the nicks to form a continuous DNA strand. E. coli DNA replication occurs in 3 stages: initiation, elongation, and termination. During initiation, the replication machinery assembles at a unique origin site. In elongation, the leading strand is synthesized continuously, while the lagging strand is synthesized in fragments. Termination occurs when the 2 replication forks meet. The replicon model explains replication initiation through 2 key elements: an initiator protein and a replicator DNA sequence. The origin of replication contains specific DnaA boxes and at-rich DNA unwinding elements. GETC sites are methylation sensitive and play important roles in replication control. 4 proteins are essential for replication initiation. DnaA binds to DnaA boxes and melts the DO region. The DnaB helicase, assisted by DnaC loader, unwinds DNA. DnaG primase synthesizes RNA primers to start replication, first on the leading strand and then the lagging strand. 3 DNA polymerases operate at the replication fork in bacteria. DNA polymerase I and III are essential, while polymerase II is not. DNA polymerase III is part of a holoenzyme complex, which includes a core polymerase, a sliding clamp, and a clamp loader. The DNA pol III core consists of 3 subunits. The alpha subunit carries out 5 to 3 chain elongation. The epsilon subunit provides 3 to 5 exonuclease proofreading. The theta subunit supports epsilon's activity but is not essential. This arrangement ensures fast and accurate DNA synthesis. The sliding clamp is a protein ring that encircles DNA and anchors DNA polymerase III to the template strand. It dramatically increases the processivity of DNA synthesis, enabling the polymerase to synthesize long stretches of DNA without dissociating. The clamp loader complex loads the sliding clamp onto the DNA using energy from ATP hydrolysis. It recognizes a DNA template primer junction and places the clamp so that it can tether the polymerase to the DNA and begin replication. The Replisome is the multi-protein machine that coordinates leading and lagging strand synthesis. According to the Trombone model, the lagging strand loops out, allowing both strands to be synthesized simultaneously. The Replisome includes helicase, primase, DNA polymerase III, and single-stranded binding proteins. Lagging strand synthesis involves 3 steps. The polymerase extends the Okazaki fragment. The clamp loader assembles a new clamp, and the polymerase dissociates, rebinds, and synthesizes the next fragment. This cycle resets with each new Okazaki fragment and requires RNase H, DNA polymerase I, and DNA ligase. The trombone model describes how the lagging strand loops out during replication to allow synchronized synthesis of both DNA strands. This looping and relooping action resets with each Okazaki fragment and enables continuous coordination of the entire process. In summary, bacterial DNA replication is an orchestrated process involving multiple enzymes and protein complexes. The replication machinery ensures high fidelity and coordination between the synthesis of the leading and lagging strands.
Cells, like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
In E. coli, DNA replication terminates when the two replication forks meet at a specific region on the chromosome called the terminus. This process is regulated by the Tus protein, which binds to Ter sites and halts the advancing replication forks. Once termination occurs, topoisomerase 4 and recombinase enzymes are responsible for resolving and separating the newly replicated sister chromosomes, ensuring each daughter cell receives a complete genome. The SV40 virus model provides an in vitro system for studying eukaryotic replication. In this system, a helicase made of 6 T antigen molecules recognizes the origin and unwinds the DNA. This helicase then recruits DNA polymerase alpha primase, which initiates replication by laying down a short RNA primer followed by a short stretch of DNA known as initiator DNA. This primer template junction becomes the starting point for DNA synthesis by more processive polymerases. Replication in eukaryotes involves a set of coordinated enzymes. DNA polymerase delta carries out the synthesis of both the leading and lagging strands. PCNA, the proliferating cell nuclear antigen, acts as the sliding clamp, holding the polymerase on the DNA. Replication factor C serves as the clamp loader, using ATP to place PCNA around the primer template junction. Flap endonuclease, or FEN1, is essential during Okazaki fragment maturation, where it removes RNA primers and associated flaps of DNA. DNA ligase follows to seal the remaining mix, completing the phosphodiester backbone. Eukaryotic replication faces several complexities not found in prokaryotes. Unlike the single circular chromosome of bacteria, eukaryotic cells must replicate long, linear chromosomes. This requires the use of multiple origins of replication to ensure timely duplication. Furthermore, 3 distinct DNA polymerases, alpha, delta, and epsilon, must work together to synthesize the genome. An additional challenge arises from the linear nature of chromosomes. Without a mechanism to replicate the very ends, important genetic material would be lost over successive cell divisions. To solve this, eukaryotic cells rely on the enzyme telomerase. Initiation in eukaryotes begins when the origin recognition complex, or ORC, binds to a replication origin. Once in place, ORC recruits additional factors, including CDC6, which helps prepare the DNA for replication. Following initiation, CDC6 is degraded to prevent reinitiation. Termination occurs when 2 replication bubbles meet and fuse. This merging of replication forks ensures that no region of the DNA is left unreplicated and marks the completion of the duplication process. 3-DNA polymerases orchestrate replication in eukaryotic cells. Polymerase alpha initiates DNA synthesis by creating a short RNA primer followed by a short DNA stretch. It does not have proofreading ability. Polymerase delta is responsible for synthesizing the lagging strand, where it works with PCNA and other enzymes to produce Okazaki fragments. Polymerase epsilon carries out leading strand synthesis, moving continuously in the direction of fork progression. These polymerases work in close coordination to replicate DNA accurately and efficiently. One major issue in eukaryotic replication is the leading strand problem. DNA polymerases require a pre-existing primer to initiate synthesis, meaning they cannot begin replication at the extreme ends of a linear chromosome. As a result, the terminal portion of the chromosome cannot be copied by conventional polymerases. This leads to a gradual loss of genetic material from the ends of chromosomes with each cell division, a phenomenon that, if left unchecked, would eventually impair essential genes and genome integrity. This slide illustrates why the leading strand cannot fully replicate chromosome ends. After replication, the G-rich strand ends with a 3 single-stranded overhang. Origins of replication are located internally, not at the very tip of chromosomes, so when replication proceeds, the 5' end of the C-rich template strand is left uncopied. In contrast, lagging strand synthesis, which uses multiple primers, can still reach near the chromosome end. The leading strand, however, cannot be extended all the way to the end, resulting in progressive shortening with each round of replication. Telomerase is a unique enzyme that solves the leading strand problem by extending the 3 overhang of chromosomes. First, a strand-specific exonuclease removes several nucleotides from the 5' end to expose a single-stranded overhang. Telomerase then binds to this overhang and, using its internal RNA template, synthesizes additional telomeric repeats. Once this extension is complete, conventional DNA polymerases can fill in the complementary C-rich strand using a standard primer and DNA synthesis machinery. This process restores the chromosome end and maintains genomic stability. The activity of telomerase is essential for maintaining the full length of chromosomes across multiple cell divisions. Telomerase is highly active in stem cells, germ cells, and certain cancer cells, where continuous division is necessary. In contrast, most somatic cells show low or absent telomerase activity, leading to gradual telomere shortening over time. This shortening contributes to cellular aging and limits the number of times a cell can divide. Proper regulation of telomerase is critical for both tissue renewal and cancer prevention. Reiterating the importance of telomerase, this slide emphasizes that by extending the G-rich overhang, telomerase preserves the complete genetic information at chromosome ends. The newly extended region provides a substrate for the normal replication machinery to complete the lagging strand. This cycle of extension and replication ensures that even the very ends of chromosomes are faithfully duplicated, preventing the loss of important genetic information with each cell cycle. As the replication fork progresses through the genome, chromatin structure must be dynamically managed. Nucleosomes are disassembled ahead of the fork to expose naked DNA for replication. Once replication passes, chromatin is reassembled to restore the structural and regulatory architecture of the genome. This disassembly and reassembly process is coordinated by histone chaperones, which manage histone recycling, and by chromatin modifiers, which regulate the addition or removal of histone modifications that influence gene expression and DNA accessibility. Visualizing this process, we see that about 300 base pairs of naked DNA precede the replication fork, and approximately 250 base pairs follow it. In this zone, histone chaperones act quickly to remove and then redeposit histones. The goal is to ensure that daughter strands not only inherit the correct DNA sequence, but also retain the proper chromatin context. Modifiers such as methyltransferases or acetyltransferases adjust histones to reestablish the appropriate epigenetic marks, enabling proper gene expression in the next cell generation. Chromatin assembly during replication is a finely tuned process that protects genome stability. If this process fails, it can lead to errors in gene expression or DNA repair, both of which contribute to cellular dysfunction and disease. Thus, chromatin reassembly is not simply about packaging DNA, but about preserving the entire regulatory framework that defines cell identity and function.
The DNA in the cell is not naked. Instead, its helical strands are wrapped around proteins called histones to form structures called nucleosomes. When you have completed this exercise, you should understand basic nucleosome and chromosome structure. Understand the process of nucleosome disassembly and reassembly. and understand the importance of histone distribution in the daughter chromatids. The human genome contains about 3 billion base pairs. If stretched out, such a DNA strand would be 1 meter long. In order to fit 2 copies of this large genome into each human cell, the DNA is neatly wound into 23 pairs of chromosomes. The first step in compacting DNA into chromosomes is the assembly of nucleosomes. Each time the cell divides, the DNA must be duplicated. After the DNA is copied, the 2 daughter strands are rapidly reassembled into nucleosomes. The old histones are carefully distributed between the 2 new DNA strands, and new histones are brought in to fill in the gaps. The nucleosome consists of a histone core wrapped with a 147 base pair length of DNA. Specialized proteins called histones form a spool or disc onto which the DNA is wrapped. 8 histones, 2 each of histone proteins H2A, H2B, H3, and H4, form the protein core of the nucleosome. This histone core is assembled from histone protein subassemblies. The H3 and H4 proteins form a tetramer subassembly. 2 H2A, H2B dimers complete the histone core. Each histone protein has an N-terminal tail. These tails provide a guide for the DNA strand to wrap around the histone core. The N-terminal tails emerge between the DNA strands and create a groove, much like that of a screw, that forces the DNA to wrap in a left-handed manner around the histone. The DNA strand wraps 1.65 times around the core, using a length of 147 base pairs. This core DNA always wraps around the histone core in precisely the same way, independent of sequence. The DNA is bound to these proteins via hydrogen bonding. The H3-H4 tetramer binds the middle and both ends of the core DNA. The H2A-H2B dimers bind the DNA between the middle and the ends. The assembled nucleosome has approximate two-fold symmetry. Adjacent nucleosomes are connected by short stretches of DNA, called linker DNA. A 5th histone protein, H1, is known as the linker histone and binds the linker DNA. The binding of the H1 histones to the DNA stimulates a first level of chromatin packing, the formation of a fiber that is 30 nanometers in diameter. The H1 histone binds both the end of the linker DNA and the middle of the nucleosomal DNA, bringing adjacent nucleosomes into close proximity. The structure of the 30 nanometer fiber has not been fully determined and may differ between species. There are 2 hypothesized forms of the 30 nanometer fiber. In the solenoid model, the nucleosome discs are stacked on top of each other, forming a helix. The linker DNA is hidden on the inside of this helical structure. A second model for the 30 nanometer fiber is the zigzag model, so named because this type of folding gives the fiber a zigzag appearance. The zigzag form requires longer linker DNA than the solenoid model, and thus may be the preferred form in species with longer linker DNA. To produce a highly condensed mitotic chromosome, the 30 nanometer fiber must be compacted into yet higher order structures. The 30 nanometer fiber is believed to fold into large loops, held together by nuclear scaffold proteins at the base of each loop. The looped fiber can then be coiled again to produce the fully condensed chromosome. When chromosomes are duplicated, the DNA must be replicated, and the associated proteins must reassemble on each daughter DNA molecule. During replication, the nucleosomes must be at least partially disassembled to allow the replication machinery to pass. The H3-H4 tetramers remain bound to one of the 2 daughter DNA duplexes at random. The H2A-H2B dimers remain intact, but are released into the pool of free dimers surrounding the replication fork. The H3-H4 tetramers from the old nucleosomes begin to form new nucleosomes on the daughter DNA molecules with which they are associated, soon after the replication fork passes. Because the amount of DNA has doubled, more histones are needed to fully package the two daughter strands into nucleosomes. These newly synthesized histones do not spontaneously form nucleosomes with DNA in the cell. Instead, histone chaperones guide these new histones to the DNA behind the replication fork. ASF1 and CAF1 work together synergistically to bring H3H4 tetramers to the nucleosome assembly site. It is believed that ASF1 helps to load CAF1 with new tetramers. CAF1 binds to PCNA rings, delivering the tetramers to the DnaA. PCNA stands for Proliferating Cell Nuclear Antigen, and this sliding clamp protein helps to tether DNA polymerase to DNA during replication. The PCNA rings are left on the DNA by the passing replication machinery, and therefore serve as markers of newly synthesized DNA. The nucleosome is completed by the addition of two H2A H2B dimers. Although less well understood, this process is undoubtedly mediated by additional chaperone proteins, such as NAP1, which is known to bind H2A H2B dimers. This process of nucleosome assembly after DNA synthesis is carefully orchestrated to preserve information carried in the histone modifications. Because the accessibility of DNA is controlled by the modifications of the histone N-terminal tails, the inheritance of this information is critical to the survival of each daughter cell. Transmitting this histone modification information begins with the incorporation of the modified old histones into each daughter strand. These modified histones are in turn able to recruit enzymes that modify neighboring new histones in the same way. Enzymes containing specialized domains called bromodomains can bind to acetylated histones and in turn acetylate neighboring nucleosomes.
In this final comparison, we examine how prokaryotic and eukaryotic DNA replication differ. In prokaryotes, replication involves a single origin of replication on a circular chromosome and is carried out primarily by DNA polymerase III. In contrast, eukaryotic cells possess multiple linear chromosomes, each with many origins of replication. Their replication is divided among DNA polymerases alpha, delta, and epsilon, with support from accessory proteins like PCNA, RFC, and RPA. Eukaryotes also must replicate through chromatin and preserve telomeres using telomerase. Together, these differences underscore the complexity and regulatory sophistication of eukaryotic DNA replication.